Okay, good evening, everyone. I think we can get started.
Good evening, sir.
Yeah, good evening. Okay, Okay, so welcome to the extra session. So basically, this is happening after the 11th week. So we're going to cover everything that has been covered in the 11th week. And for 12th week anyway, we are going to have that extra session next Tuesday. Okay, so what I've tried to do is basically uh, try to cover everything from start. So I obviously we cannot cover each and every detail because to our session it is not possible. So I have kept the topics I think are like more necessary to understand. I skipped a few details here and there, but I had covered all those things in the live sessions for those weeks. So I don't think that should be a problem. So if I've missed anything here and you think that it would be better if I cover that topic as a revision topic in the next class, I'll be happy to do that. Please let me know if I missed something that you want me to cover. Okay, we are not going to discuss any problems or anything of that sort because I don't have any assignment problems for this week uh, for this extra session. What I'm going to do is discuss basically the basic concepts, the ideas, uh, and try to reinforce the understanding of these concepts. That's all. Okay, so let's get started. Okay, so at the very basic, I'm going to start with the very basics. So Uh, I hope all of you remember genetic codes, how, firstly, the central dogma of biology, DNA to RNA to proteins, and how DNA is converted to RNA and to proteins. So DNA replicates itself, then it's transcribed to RNA, and then it translates to protein. So RNA to protein translation is aided through through reading of these codons, which are these three-letter words, three-letter sequences of nucleotides and basically other nucleosides in the RNA. So in this case, in the genetic code, since thymine is not present in RNA, thymine is replaced by uracil. So you see A, C, G, and U instead of T, and each of these three-letter codons code for a particular amino acid. But there is a lot of redundancy because there are 20 amino acids that our body can code for, or our DNA can code for, and there are like total 64 possible codons. So there is a lot of redundancy, and that redundancy is due to evolutionary constraints because some amino acids, if something, if an if it's an essential amino acid to be made by the cell, and some sites of the codon is mutated, at least there will be some other codon that can code for the same amino acid. So evolution wise, it makes much more sense to have this redundancy, right? Okay, so there are two sets of uh, specific codons which do not code for amino acid. One is the AU, it codes for an amino acid, but it's the start codon. AUG is the start codon which codes for the amino acid of methionine. It's always the first amino acid. And there are three stop codons, UAA, UAG, and UGA, which when the tRNA, sorry, when the uh, when the polymerase enzyme, which is translating uh, the mRNA to protein with the help of tRNAs and other types of RNAs, when it encounters the stop codon, it stops the transcription, and the fully transcripted um, sorry, stops this translation, and the fully formed peptide length of sequence is let off from the complex, right? So uh, now, as I was saying, that there's the start codon, but uh, from the start codon to the end codon, everything that falls within it is known as an open reading frame, right? So there are, each of these are starting off with AUG is giving a particular amino acid. So AUG codes for methionine, and then this will code from some other amino acid and some other amino acid, and so on and so forth until and unless it reaches the stop codon. So the ribosome will fall off when it sees the stop codon, and the tRNA falls off basically. So this is the open reading frame, right? Where from the start codon to the first end codon, stop codon, what is the sequence? So that is the open reading frame at any given location in the genome, right? So we have discussed a little bit about DNA nucleotides, ATGC, and how basically some of the two classes, you have purines and the pyrimidids, and we have also discussed the structure of the DNA a little bit, where you have this 3D double helical structure, where at every groove you have around 10 bases stacked on top of each other in a ladder form, and it goes up to around, I think, uh, 10 angstrom, right? So there's a minor groove and a major groove, and all the structural details about the DNA I'm not going to go into details of. I hope you remember all those things. But we also discussed this thing, which is the stacking energy. So if one base pair is getting stacked uh, on the other, what is the energy that is formed, or if there are basically overlapping what we do is we find these overlapping nucleotides of two sequences. So if it's a given sequence, we find the overlapping two base pair sequences, so AC, CT, TA, and so on and so forth. And then we retrieve the energy values associated with each of these yeah, dinucleotides, right? So that there are already established ways by which you can find this stacking energies, and most of these are already found and stored in databases and standardized. So we find these possible pairs, we retrieve the delta G. Yes, some question. Okay. Uh, yeah. Uh, yeah, and basically for each dinucleotide we get the delta G, and we sum it across all the possible dinucleotide sequences that we have, and we normalize this with the total number of dinucleotides, which is going to be n minus 1 if n is the length of the sequence. So this is the formula, which basically you are summing up the individual pairwise delta G's, and you're dividing it and normalizing by the length of the sequence. So I covered this because there were some questions in assignment problems related to the space stacking energy. So some might appear in the exam, so that is why I have covered this. This might seem a little out of place with the rest of the flow of the presentation, but yeah, I thought you might want to have a recap of this because this might come in the examination. Okay, next, at the next level, we have talked about different types of biological databases. We have discussed most of them, but more details for the protein related databases where you have protein information. You have also specific protein databases where you have uh, structural information like PDB and all. You have nucleotide databases and genomic sequence databases as well, which we have not touched up that much. But yeah, there are a lot of uh, since data is getting generated so fast, so we have all these databases uh, that we can get the data out of. So basically, these are the classes of the databases, 7 classes, and these are the information that it provides. I hope this kind of summarizes all the different kinds of databases that we have. Right, so PDB would be some forming in terms it will be falling in terms of protein databases, and if you have some comparison matrix, so for example if you are doing cat or some multiple sequence alignment or something which related to different proteins, and the information that you are getting in terms of protein families, how two proteins are similar or not, if they're part of the same family, you are same sharing same domains and functional sites. So those are different set of databases uh, which you have, like cat is one such database like that. So yeah. Okay, so yeah, so this is one type of categorization on the basis of the information that is present in the database. The other type is basically the level of information it is present. So in the primary database, you will have primary level information of the DNA or the protein that you're trying to find. So we have sequence databases and structural databases. Then we have secondary databases which has related information to the primary database, right? So if you have a structure, if you have a structure and you are breaking down the structure into different domains and you are studying each of the domain and putting some information of the domain in a database, that becomes a secondary database, right? And other kind of databases would be like pathway database, where you have information about multiple genes forming a particular pathway. And so basically, it's not the primary information, the gene information will be present in some primary database like NCBI, RefSeq or something like that. But the secondary information in level of pathway levels, say for example, is present in cake pathways or MSigDB and so on and so forth. Then we have the composite databases where you have both primary and secondary information, and others where you have literature-based information or other kind of other non-human information or so on and so forth. Okay. Okay. Now, these were, I think, we are coming to the end of the first session that we had. The basic topics that were covered. I also talked a little bit more about uh, the different uh, different amino acids and their functions and their basically chemical properties and all of those. I'll cover a little bit on that. But yeah, that is mostly that what was covered in the first session where you had introduction and a little bit of exposure about different kind of databases and how different kinds of data is stored across databases, right? So next we focus more on the proteins. So the most important part of the protein is a peptide bond, right? So protein is a long uh, length of peptides in a sequence connected one after another. The connection between two amino acids is the peptide bond, right? So this is the peptide bond. The peptide bond is formed with respect to the carbonyl group, which is the CO group, and the amide group, which is the NH group. And the bond is formed between the carbon and the nitrogen. And this is the carbon that forms the bond with the nitrogen is known as alpha carbon. And with respect to the alpha carbon, we define different positions in the amino acid as well. Okay, so these are the different categories of amino acids that we had discussed. So basically, you have mostly four or like you can say three, where you have hydrophobic amino acids, where these do not have any polar side chains or any charge associated with them. So these are hydrophobic amino acids which will be buried inside the structure of the protein within the inside residues because it does not want to come in contact with water and so it is always going to be buried inside. On the surface of the protein, you're going to find the more polar and the charged residues. There can be positive charged and negative charge. These charge also help in stabilizing the protein with electrostatic bonds when the secondary and the tertiary structure forms. But also these charged proteins are also charged amino acids are also going to be found on the surface of the protein as well when the tertiary or the quaternary structure of the protein is formed. So one thing to remember is basically the three-letter symbols. Please at least try to remember the major ones in terms of glutamic acid and glycine gets a lot of kind of confusions. Leucine and isoleucine, leucine and lysine, these kind of three-letter codes are very similar and you might get confused. So in the exam, you might probably will get questions for topics like this with respect to three-letter amino acid codes, and it's not going to give you the whole name of the amino acid, most probably. So remembering the three-letter amino acid codes are important. So please do that. And aromatic amino acids are basically another class which do not have charges or are not hydrophobic, but they have this this side chain, which is a benzene ring or uh, some sort of ring-like structure, be it a single ring or a double ring. So tyrosine, tryptophan, phenylalanine, and histidine are the four major groups of these aromatic amino acids. Okay, okay. So peptide, we talked about different amino acids in a peptide. So it's a long chain of these amino acids. Now, that is known as the primary structure, which is just the sequence of the amino acid, right? On top of that, the, the peptide will fold, form secondary structures, which are mainly alpha helices and beta sheets. Then you have tertiary structures, which are also more governed by the different van der Waals forces, the electrostatic forces. If there are sulfide bonds, those are going to help. And finally, non-covalent association of multiple protein subunits. If it's a multi-subunit protein like a histone or something like that, you'll have more interactions between these individual subunits and which will form the whole protein, right? So primary, secondary, tertiary, and quaternary. Primary is the sequence. Tertiary is secondary is one step ahead of it in terms of alpha helices and beta sheets. Tertiary structure is folding of these alpha helices, beta sheets into different structures to form a protein subunit. Now that subunit hence a protein is made up of multiple subunits. So subunits are going to come together and make the whole protein, which will be the quaternary structure. Okay. Okay. As I was saying, for primary structure is basically the length of the sequence of the amino acids in the peptide, uh, in the peptide, and basically what we are looking at are basically amino acid sequences, disulfide bonds, because this cysteine and methionine, they have the sulfur content, and basically they can form these disulfide bonds. We have proteolytic cleavages also that we can look at, because so when you're trying to break down a protein or break down a peptide bond, we need some enzymes to break the bond between the carbonyl carbon and the amide nitrogen. So yeah, if you are looking for those things, primary sequence is something that becomes very important. And also we can have post-translational modifications on the primary sequence as well. So when from RNA to protein the translation occurs, after the protein is getting formed, after the peptide bond peptides sequence has get formed, you can add multiple post-translation modifications on the protein, which can help the protein fold, or also which can tag the protein in terms of where the protein needs to go. So if, for example, um, you have the protein that is formed is mutated and it's ill-formed, and you need to destroy the protein. The cell needs to destroy the protein. So what it'll do is it'll tag it with a ubiquitin tag, so that the Golgi and the other uh, uh, organelles in the cell can identify the tag and basically destroy the protein, or basically transport it out of the cell. So yeah, majorly these things that we look at in terms of the primary sequence. In terms of the secondary structure,
Sir, I have
oh
a
yeah,
question
yeah,
in the
sure, sure.
slide. I have a question in the slide. It was written primary structure and then disulfide bond, but I think the disulfide bond are referring to tertiary structure, right?
Yeah, it can
They are not
definitely.
e-
No, no.
significant.
Yeah, definitely. Uh, in tertiary structure, the function of the disulfide bonds are to stabilize the tertiary structure, just like other electrostatic bonds and other covalent bonds that we see in tertiary structures. Here, what we are talking about is if you have two of these cysteine or like the sulfur-containing amino acids side by side, what can happen is instead of forming, other than forming the peptide bond, it can also form these secondary bonds, which are you're going to be sulfur sulfur bonds, right? So these are you can definitely see them in the primary structure itself. So here we are talking in terms of that in primary structures, in primary sequence, other than your normal peptide bonds, you can also see some sulfur sulfur bonds if sulfur containing amino acids are present next to each other. In the primary structure only, it can form those bonds. In the tertiary structure, what can happen is if you go, it'll the the sulfur sulfur bonds will be formed over long range contacts. Over after folding, if two of the sulfur containing amino acids come close to each other, but which are very far away from in the primary sequence, they can form a bond there. But if two sulfur containing amino acids are sitting side by side, there's a chance that it can form a sulfide bond. So that is what we are trying to say here. But like, yeah, if you're getting confused,
Okay, sir.
forget about it.
Ah,
Just focus on the primary bond.
yeah.
Yeah.
Okay, sir. Thank you, sir.
Okay. Uh, in the secondary structure, yeah, as we're saying, it's 1 second. Yeah. Uh, so basically, what happens in the secondary structure is there's some resonance between this particular set of bonds. So what happens is you have a double bonded carbonyl here, and you have a single bonded amide here, and basically this double bond can resonate to form a double bond here, which will be so in terms of resonance, neither this bond or this bond will be single or double bond. It will be somewhere between single and double due to this resonance, and it shortens the bond length in with respect to a single bond. The single bond length will be higher. The double bond length will be much lower. It somewhere sits between the single and the double bond because of this resonance that gets formed with respect to the carbonyl carbon and the amide nitrogen, right? And so we have different types of secondary structure, which is the alpha helix and the beta sheets, and we have different. So we are going to discuss the Ramachandran plot a little later, but we have different constraints on these secondary structures about what is the what are the conditions under which it can form a primary alpha helix, or and what are the conditions under which it can form the beta sheet, right? So this condition is determined by two angles. One is psi and the other is phi. So one is, I'm going to show you that in the next slides, but basically the these bonds and these bonds basically. So the angle between these bonds will tell you how much like what is the probability that it can form a alpha helix, or what is the probability it can form a beta sheet. So yeah, we were going to discuss the Ramachandran plot a little later, but alpha helix generally is right-handed in nature, and it is basically a little bit more common than the beta sheets, but yeah, both of them are very well studied. Okay, so yeah, so basically this I'm going to skip this because I'm going to take this up in the Ramachandran plot discussion when we are going to have that. Okay, so in the primary structure, we just have the sequence. Secondary structure, you have the alpha helix and the beta sheets. And in the tertiary structure, what you form are basically uh, almost the final set of the structure of the protein, unless and until they are multi-subunit proteins where you need to form bonds with other subunits as well. So mostly, if it's a single subunit protein, tertiary structure is the final structure that we're looking at. And basically, the whole structure of the protein determined uh, uh, will determine the function of the protein as well. So you can either have globular or fibrous proteins. So globular proteins will have a different type of functionality. Fibrous protein mostly present in muscles and these kind of things, collagen and all these things. So depending on the structure of the protein, the functionality also differs, right? So and these globular proteins are like ball-like structures, having ball-like structures, and they do not have very elongated structures, right? So uh, most of the non-structural proteins, including enzymes, are globular. Where you need elongated structures are basically your skin, keratin, your connective tissues, your collagens, your basically muscle structures. There you need larger physical contacts between 2 points where you require these long fibrous proteins. So keratin is a good example of a fibrous protein. Collagen is also a good example of a fibrous protein. Any kind of enzymes are good examples of globular proteins. So in this case, it is shown as lysozyme. Okay, and finally, quaternary structure is basically if you have multiple subunits, or you need something else other than the protein to stabilize the protein, you fuse or you kind of form bonds and attach to the other subunits to form the quaternary structure. And basically, it's a similar set of forces that are acting on the tertiary structure. We're going to discuss the forces a little later. So, and they can be mostly homomeric or heteromeric. And one good example of that would be hemoglobin, right? So, hemoglobin has I think, four subunits, if I'm not wrong. So all of the subunits are generated separately and then they're merged together to form the whole hemoglobin molecule. So this is how it looks. And you have, I think, two subunits shown here, which is one is the pink one is the yellow one. Uh, Okay, so now we are going to talk a little bit about what can we do with the primary structure, that is just the sequence of the protein. Once you have the sequence of the protein, what you can do is you can compare the sequence across other protein sequences, right? Be it from the same organism, same species, or from other species. So the idea is that if you have similar sequences, it shows an evolutionary relationship, and there is something that is getting conserved across generations. So, for example, if you're taking I'll take the example of histone again because it is very common. So the histone protein is basically one of the most conserved proteins across organisms. So if you check the alignment and basically the similarity of the histone of a human and with the histone of some ape or some chimpanzee or monkey, it is going to be very similar. And not only mammals, if you check it with other eukaryotic organisms as well, it shows a very similar sequence because it is a very fundamental protein involved in DNA packaging, right? So it is involved. The DNA packaging has not evolved across generations, so that's why the histone protein has also not evolved across generation, and it is at a stable point and it does not need to evolve. So if you have a similar sequence that is getting conserved across generations, across species, that means that protein is very important, and the function that it is doing is very fundamental to the existence of the organism. Right? And if you have same proteins which are very different in terms of sequence between two common ancestors, what you can say is basically the function of the protein has diverged during the evolution of these of the daughters or basically the next generations, right? So if you have a similar sequence, there's some evolutionary relationship with respect to that. And if the sequences are similar, it is also the functions are also related in most of the cases. So these are what are known as orthologs. So they have almost same function in different organisms and almost similar sequences as well. Right. So now you want to quantify these sequence similarities. So there are different ways to do it. The most simplest way to do it is using dot plots. And basically, if you have two sequences, you make a 2D plot where you put a dot if a particular amino acid at a particular position is same, and you leave it blank if there is a mismatch, right? So this is how it looks like. So this is with respect to nucleotide sequences, but it's the concept is same as of protein sequences, amino acid sequences as well. So if two bases are matching at a particular point, we put a dot. Other than that, we leave out all these blanks, right? So a lot of things can be told in terms of the dot plot uh, type that is getting generated when comparing two sequences. So if it's a diagonal like this, on the leading diagonal, if you have more points, that means the two sequences are very similar. If in the off diagonal you have a lot of points, that means the two sequences, one is a reverse of the other. And if there are gaps between that, those could be mutations, those could be some other variations as well, right? So this is uh, uh, dot plot, how a dot plot looks like. And if you have a good relationship, a good sequence similarity between the two proteins or the two sets of DNA that you're looking at, this would be one line and not so scattered along. So if it is so scattered along, it is telling you that a lot of matches are there between different regions of the protein, but none of them are in sequence, right? So this is not a obvious sequence similarity between these two proteins, right? So this is not a good one, a good similarity. Uh, so particularly this plot is not showing any good similarity between the two sequences that it is comparing. Okay, so yeah, as I was saying, I'll not waste much time over here. So if you have having very similar sequences, you get a get a line on the leading diagonal. If you have inverted sequence, the line will be on the off diagonal, right? Okay. In terms of the alignment, it's basically you're checking two sequences and asking if how much similarity is there between sequences. In terms of quantification, if you want to do so, so you have a sequence and you want to align a sequence to that. So there can be many ways to do that. So we need to have a scoring metric for that, right? So there can be multiple ways to have good alignments. We need to have a scoring metric which will tell you that this is the best alignment if you have the maximum score. Right. So the most basic score would be if there's a match, it's one. If there's a mismatch, it's zero. So again, it is not very robust because uh, the mismatch can also be different. So if it is a mismatch between similar amino acids, that does not mean that the protein is drastically different, right? But there's a mismatch. If there's a mismatch between two different kinds of amino acids, say for example, a polar amino acid has been replaced by a hydrophobic amino acid, then it becomes an becomes a bigger difference than a polar amino acid being replaced by another polar amino acid. So this scoring technique is not very rigorous. So what we do is we also want to penalize. So first, though, is that that thing is that not all substitutions are having the same weightage. And second thing is if there are gaps, you need to penalize the gaps because those are mutations. So again, if what kind of gap penalization are you going to do? If you have same penalty for each of the gap, then you can get something like this on the right? But it is not. If you compare these two alignments, the one on the right is less biologically feasible than on the left, because if you are having a mutation, it is very hard to have a mutation one base pair left and one base pair right, and not have a mutation in this one. So if there's a gap, the more probability is that that gap will get extended rather than it'll form a gap something a small base pairs down the line, right? So this is improbable than this, right? So from a biological perspective, from an evolutionary perspective, we use gap penalties in a different manner. So what we do is we we use a gap opening penalty and then we extend the gap penalty depending on how many gaps are there. So and the gap opening penalty is much more than the extension penalty. So it is very for the alignment algorithm. It it will have to penalize a lot if it is doing an alignment like this versus like this, because here there's only one gap opening penalty. In this case, there will be two gap opening penalty, right? Yeah, and I was saying that not all uh, substitutions will be the same in terms of evolution, in evolutionary impact. So we need to have some metric to quantify that this substitution is less impactful from evolutionary perspective than that substitution, right? So we build scores, build scoring metrics, matrices which can tell you all different combinations of substitutions and what is the relative score, uh, right? So we create this mutation data matrix, which is a scoring matrix compiled based on observation of protein mutation rates. Some mutations will be more observed than the other, and basis of that, they have developed scores and these metrics, which is known as PAM matrix or the Blossom matrix. So these are two different uh, scoring metrics for amino acid and protein sequence alignment. Okay. So this is basically a little bit of information about how the PAM matrix is created. So here PAM is basically a unit of time needed to acquire one mutation per 100 positions. So basically, in how many generations one mutation can be formed in 100 positions in that region of the protein. So that is what PAM is determining, and the model followed by it is kind of this. So you have, so this is for DNA, but it's similar for proteins as well. So you have different probabilities of transitions and transversions. So transition would be purine to purine, pyrimidine to pyrimidine, and transversions would be the purine to pyrimidine or pyrimidine to purine. Uh, the similar or analog to protein sequences would be polar to polar, polar to unpolar, and unpolar to polar. So uh, different different amino acids getting substituted compared to the one there. So, for example, a polar is getting substituted by a hydrophobic amino acid residue. So the structure around that will change much more compared to if a polar is replaced by another polar amino acid. So if you know assume equal probability for each mutation, it becomes this is the mutation matrix. It becomes, and for Duke's Cantor model, they assume that the alpha and the beta, so probability of purine to purine and pyrimidine to pyrimidine transitions are the same. It considers the same, and for the Kimura model, it considers slightly different. So purine purine is less, and purine to pyrimidine is more. Right. So, and when you take all these observations, ki how many uh, mutation rates are possible. So basically, if you take the probability of observing amino acid B in the position in a particular position, so you derive this particular metric, which is the odds of seeing a substitution as a result of mutation versus seeing it by chance. So you have a background distribution, and you have a probability of the actual mutation rates, and then you can make a scoring function like this, which will be a log score, uh, and it is what used for a framework like this is used for creating this PAM and BLOSUM matrices. And as I was saying, PAM is basically point accepted mutation, and if you're saying that two sequences are uh, defined by one to be one PAM unit divergence, is basically it can convert from one sequence to another with an average of one mutation per 100 residues. So what is the sequence similarity is what PAM or BLOSUM is telling me. Okay, so specific class, I've included this couple of slides here because questions might come regarding the notation of enzymes, right? So enzymes are a class of proteins that are very important for the cell because it acts as a catalyst between every biological reaction that happens within the cell, and we have different naming systems for these enzymes. There are majorly seven classes of these enzymes, as the name suggests their function. So oxidoreductases will do either oxidation or reduction. Transferases will be transferring some group of say for example kinase. It will transfer a phosphate group from one molecule to another. We have hydrolases which can help in uh, uh, which basically will help in react to water, and it will add water molecules to the compound itself. You have lyases which break down molecules. You have isomerase which converts the compound into its isomer. You have ligases which ligates or like joins two separate molecules, and you have translocases which basically transfer one uh, 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 one group of compound to another group. Basically, you're changing uh, the the composition of the protein or the compound that you're working with. So the important thing is here is basically the naming system. So you have these seven major classes of naming systems. So EC1 to EC7. So each of these combine give sorry each of these is corresponding to a particular type of these enzymes, and each of these have some trivial names as well. So it can come as some trivial the question can come as some trivial name as well. So kinase is a transferase because it is transferring phosphate enzyme, phosphate group. Isomerase, as I was saying, makes an isomer of the compound that we are working with, transporters and synthase and so on and so forth. So just kind of remember how it is named in terms of IUPAC. So basically, if it is given like this, right? So EC, there will be majorly four numbers separated by dots. So the first digit will tell you the main enzyme class, which will be anything of these seven. The second will be basically the subclass. So it will determine what specific type of reaction within the main class will the enzyme be involved in. The third will be the subclass beneath that, which will be the reaction mechanism or the specific type of chemical transformation the particular enzyme is doing. and the fourth and the final one is basically the unique identity of the enzyme that catalyzes a reaction within a given class. So if you just give an EC3, it's just a hydrolase. 3.4 is a specific hydrolase that will act on a specific um, type of bond. 3.4.11, it will be a peptidase that will cleave the first amino acid. So that's more specific. It'll act. The second one is acting on peptide bond. Third one is acting on peptide bond at which position. And fourth one is basically it'll remove. It's the very specific case where it'll remove the first amino acid from a tripeptide. So basically, the naming convention is important. So I thought I'll cover this. Okay. Okay. So okay, now, so we were discussing about alignment and finding sequence similarities between two set of sequence. So there are two types of alignment that we have majorly talked about. Is first is the global alignment, which is comparing the two sequence end to end. Is basically you're aligning the entire sequence of one with the entire sequence of the other. uh, and mostly it is good if you're doing same length, right? Because if you have two vastly different lengths, global alignment does not make sense. And in local alignment is basically if you have two very differently sized sequences and you think one sequence is a subset of another sequence. So here, instead of trying to align the entire length of the sequence, only regions with the highest matches will be aligned. So it will be helping. So if you have a conserved region in a particular protein, and you want to align that with and see if those conserved regions are also conserved in other kind of proteins. So you are going to only have the sequence of the conserved region, and you have large protein sequences of the other proteins. So in those cases, you're going to do local alignment rather than global alignment because it's not end to end. Right. So this is a schematic representation of global and local alignment. So yeah, hope it is clear. Okay. So now in terms of how many sequences you're aligning, there can be two different types of alignment problems. One is pairwise alignment. If you're just doing, if you're trying to align one set of one sequence to another, so that is pairwise, and it can all try to align multiple sequences together to find a particular region which is conserved across those multiple sequences. So those will be multiple sequence alignment, which is three or more sequences that you are trying to match. Uh, so for pairwise alignment is basically uh, the the purpose would be to find the degree of similarity between any two set of sequences. For multiple sequence alignment, the idea would be to find functional and regulatory or like conserved regions across different species in the same sequence, right? So that would be for multiple sequence where you're trying to see a particular region across multiple sequences. So that would be MSA, and if you're doing this too, it will be pairwise, right? So for pairwise alignment, we have different algorithms, as I was saying, that you can have. So all these things that we discussed, uh, the identity scores and the gap penalties and all these things, we're talking with respect to uh, pairwise alignment. And if in pairwise alignment, so we have different algorithms to use this mathematical framework and give the alignment and give a score out of that, because computationally it is going to be very difficult if you are trying to align a sequence of length 20 to a sequence of length 2 million. So it is computationally very difficult. So we need efficient algorithms to do that, right? So dynamic programming is one such efficient algorithm, and it is uh, it is quite widely used, and it's quite well established, right? So what it does is it is basically divide the whole sequence problem into small sub problems which can be solved only once, and in parallel with each other and store the value and then ultimately uh, converts them into something. So in this case, for a particular position of alignment, there can be two cases. So it can align, so Si minus 1 and J minus 1. So you're trying to align Si with Sj, right? So it can align Si minus 1 and Sj minus 1 and add the alignment score of Si and Sj. If there is a match, what is the alignment score of that? Or so basically in the first case is the perfect match, right? In the second case is basically I is aligning with J minus 1 and there is a gap. Sorry, there's a gap in between. So to account for the gap, and also there is the third possibility where I minus 1 will align with J and also there will be a gap. So across all these three, which is the best possible alignment in respect to the score will be selected by the algorithm. So the algorithm reads like this. It will be the maximum of the three, which is the best, which is going to be the best alignment at that position. So yeah, as I was saying, so you're move, so this is the ith position and this is the jth position. So if I matches with j minus 1 here, so this is one possibility. So I and j minus 1, the green one, uh, I minus 1 and j minus 1 will be this point, and and I minus 1 and j will be the blue point, right? So you can arrive, so you're trying to arrive at this particular alignment, right? One I and j. So you can arrive at it from three different angles, and whichever is giving you the best score will be the optimal alignment at that particular location, right? So yeah, I'm going to skip these. Basically, these are talking with respect to time and space complexity. So I'm not going to go into details of those, but these PPTs are going to be there if you have uh, anything to, like if you want to go back and read these are always going to be there, and if you have any issues also you can let me know. I'll try to explain. Okay, so yeah, as I was saying that you can arrive at this particular possibility of alignment from three different angles, and basically the maximum output, the maximum score across the three, which one will give will determine what is going to be the alignment there. Okay, so yeah, and basically after you have done this alignment, so for every possible location you're going to do this thing, so basically this thing for every possible location, and ultimately you're going to come up with scores across every possible combinations. So the idea is that you start, so after you have created this matrix you need to recover the path or recover the actual alignment, right? So what you do is you start from the end and then you move. So if you start from here, there are three possibilities of moving, right? So you can move top, left, or right, or diagonally or left, right? So you will move where there is the best score. So if you start from here, if the best score is here, you move here. Here also you have three possibilities: left, top, and diagonal. In this case, the diagonal has the highest score, you move here. Then from here again, three possibilities. You can move here, 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 right? So again, I think the left one has the largest one, then you move. So these arrows are telling you basically where uh, from where you're moving to where, and the color of the arrows are telling you if it is a gap, it is a match, or it's a miss. So if it's a gap with respect to this sequence, or it's a gap with respect to this sequence, or if it's a match. So this one, the last green one, it's a gap with respect to the first sequence, the top one. So yeah, so that is there, and this particular alignment is telling you that there's a gap with respect to the second sequence. So yeah, so basically you create this dynamic programming table using this algorithm. You create this and you create the whole thing, and from there you have to recover the alignment. So you start from the end and start moving towards where the highest score is. If it is possible that you can have multiple, so in this case, so for example you could have this to be the same score as well. So if you have multiple possible ways to go with the same high score, that means there is two possible alignments, or there are multiple possible alignments which are giving you the same score. So the path need not be unique is what the idea is. Okay. uh, Uh, yeah, I'm going to skip these as well. These are like more details with respect to the dynamic programming, which I'm not going to go into much details of. I think I had explained in in the uh, in the session in which we had covered these things. Okay, so yeah, and yeah, also I do not want to make you get more confused regarding all these things because I don't think there is going to be many questions regarding dynamic programming and those kind of things in your exam. So focus on the basics, focus on the for understanding the basics. I think that would be good enough. Okay, so again, this slide is showing a basic difference between local and global alignment. So yeah, uh, we've discussed this. So it's a small stretch of alignment in a small stretch of the region, and the global alignment is basically it is across the whole sequence of the two ones. Now, uh, dynamic programming we saw for two sequence alignment. So right, so this is like two sequence you're doing the alignment for. You are using dynamic programming, but for multiple sequence alignment, so uh, you have to consider exhaustive all possible alignments at once, right? So what you do is you extend this dynamic programming algorithm to a multimodal, multidimensional matrix. So in this case, you had only one matrix for comparing two sequences. So what you're going to do is basically if you have four such sequences, there will be three groups, right? A, B, B, C, C, D, and D, A, right? So there will be four possible pairs of alignment. So you're going to do this four alignments separately in different matrices, and then you are going to combine all of them and get the dynamic algorithm score for that. So what they do is basically they use instead of a single matrix, they use multidimensional search matrix. Uh, so if you have n sequences to align, there will be n dimensional matrices required, and so and there also you use dynamic programming. But the amount of the issue is that the computational time and space is very high. So uh, if you have typically shorter length of sequence and less number of sequences to handle, then only use dynamic programming. Other than that, there are some other sophisticated algorithm that can be more, or there can be heuristics that can be more efficient as well. Okay, so one such alignment algorithm is BLAST, right? So BLAST is one of the most common of these algorithms, and there are like different types of BLAST. So if you are comparing protein sequences, there is a different BLAST, which is BLAST P. If you are comparing nucleotide sequences, there is BLAST 10. If you are comparing a nucleotide sequence query to a protein sequence database, that is also possible, which is BLAST X, and the other ones are also there, right? So Blast works in a very simple manner. So what it does is first it pre-processes its own database, right? So you have a lot of sequences inside the database. So what it does is it breaks it down into small sequences which are known as k-mers. So the k would determine the number of sequences that you are working with. So in case of nucleotide sequence, it's generally 11 sequences that they consider. So the number will depend on the how efficiently you want to index your sequences and so on and so forth. So if you have larger sequences, your k should be a little bit larger. If you have shorter sequences, the k should be a little bit shorter, right? Because nucleotide sequences will be much larger than protein sequences. That's why you have um, 11 mers in case of nucleotides, and you have 3 mers or 4 mers in case of protein sequences. Right? Okay. So this is the first step. It breaks it down into smaller, shorter segments. And in the second step, what it does is basically it searches a database of known sequences to find any sequence that contain the exact same words as the query sequence. And this is done to identify database sequences containing the same sequence, right? So in the preliminary search, what it does is, so if you are given a sequence, it is broken down into smaller k-mers, and these smaller k-mers are used for searching in the database. And in the database also, it is the nucleotides are broken down into k-mers and stored as indices. So the k-mer to k-mer search becomes much more feasible than searching a whole 200 base pair long nucleotide across a 3 billion longitudinal rate. So it is much more simple computationally. And finally, then it gives a similarity score between the matching k-mers, right? So the matching of the words is given by a substitution matrix. And if a word is above certain threshold, it will be considered as a match. Otherwise, it will not be uh, considered as a match. And it uses known substitution matrix for protein sequences. It uses PAM and LOSUM. For nucleotide sequences, it's match mismatch scoring. So if you have a match, there is something. If you have a gap, there is something penalty. And if there is a mismatch, depending on if it's a purine-purine mismatch or a purine-pyrimidine mismatch, you have something, some other score for that as well. Okay. And finally, the fourth step is involving the pairwise alignment, and it's tried to extend on both sides while counting the alignment score on the same substitution matrix. And so what it gives is basically a segment pair without gaps, which is known as the HSP or the high segment pair. And it also gives a value, a statistical significance, significant value, which is called the E value or the expected value. So E value represents the probability of obtaining a sequence match by random chance. So if E is lower, the more significantly matched is your alignment. If E is higher, then it is more likely that the match has been due to random chance rather than actual similarity. So E value will give you a statistical significance based on the alignment. Okay. Yeah, I have given some more details on the E value, but yeah, if you're interested, please take a look at that. This is anyway. I'm going to take a 2 minute break. I'll just be back. Mm, okay, sorry. Mm, I hope uh, I'm audible, right? Can anyone give me a thumbs up? Thanks. Okay. Yeah. So uh, yeah, we are talking about BLAST, and basically the statistical significance score that it gives along with it for uh, for sequence alignment across multiple sequences or two sequences at least. Okay, so yeah, again, I'm not going to go into details. These things are not going to come in your exams, but if you want to know how E-value is calculated, so this is the formula. There are some parameters involved here. So the lambda and the k, negative lambda here and k here. These are some parameters. They are estimated from large sets of random sequences, and uh, the lambda parameter will normalize the alignment score, and the k parameter will scale the e-value based on the database and the sequence length. So yeah, if you are interested in e-values, please look into this slide. Okay, now we are going to talk about, so we, what did we cover in the last hour? We talked about first amino acid sequences, which are primary structures, then a little bit about different protein structures. Then we talked about sequence and what sequence alignment means, different methods for doing sequence alignment, BLAST as a tool for doing sequence alignment. Now, the idea that we are trying to look at is like the different types of, of features of the protein in terms of the structure and the chemical function, not the sequence similarity, right? So sequence similarity is one way of looking at different types of proteins. The structure and the chemical properties is another way of looking at this protein. So one such chemical property is hydrophobicity, which is basically the repulsion of water, and it's one of the major driving forces of protein folding. So you can, so in terms of being hydrophobic or non-hydrophobic, you can have hydrophobic residues which will be very deep inside the protein core where it is far away from the water molecules, and you can have charged or polar molecules, polar amino acids on the surface, which will interact with the water molecules, which will help help the solvent solve the actual solute, which is the protein itself, right? So arginine is a well-known polar amino acid which is strongly hydrophobic, and it's present on the surface of these proteins, and it helps in solubilizing the proteins itself. Now, quantifying hydrophobicity is important because we want to compare hydrophobicity between different amino acids and try to understand protein structures based on that. So there are different scales of hydrophobicity. One is tied to little scale, which is the most commonly used. This is based on where residues are actually found in the crystal structures. So basically, if you have positive values for tied to little scale, it will be hydrophobic. And if you have the more positive, the more hydrophobic it is. So alanine is hydrophobic, cysteine is hydrophobic. If you see arginine is highly negative, which is opposite spectrum of hydrophobic, and so on and so forth. And there are other uh, 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 types of scales as well, but we have only covered the kite to little scale, so I'm not going to go above that. But yeah, so there are different kind of scales possible, but for kite to little, the more positive it is, the more hydrophobic it is, and the more negative it is, the more hydrophilic it is. Okay. uh, so this is basically calculating hydrophobicity with respect to a, how do you calculate this hydrophobicity with respect to a window of amino acids, which is a stretch of amino acid, because hydrophobicity will be depending on that the protein will fold, right? And it will not be depending on one amino acid, it will be depending on a stretch of amino acid. And what we make out of that is basically what is known as a hydropathic plot. So across a sliding window region, we calculate, we plot the hydrophobicity values of the amino acids in that window, right? So basically, it is averaging the hydrophobicity in a particular window. So in some cases, some areas the hydrophobicity will be higher. So in this case, probably it will form the helices where the turns. Uh, then you will have regions of lower hydrophobicity which will form the loop, and so on and so forth. So basically, in a transmembrane protein where you have a structure like this, a structure that goes inside the membrane and outside it, so the the amino acid presence inside the membrane will be hydrophobic definitely with high hydrophobicity score, and outside the membrane on the loop will be your hydrophilic uh, amino acid residues. So this is what is known as a hydropathic plot. So you have a sliding window and you sum up the hydrophobicity across that sliding window, and you can find these distinct regions of hydrophobic and hydrophilic amino acid residues together. So it can tell you, inform you about how the protein might fold. So this hydrophobicity is good for protein folding, but in many cases it is seen that you can have both hydrophobic and hydrophilic residues uh, being in very close proximity to each other. So in alpha helix, you can see this is the structure of an alpha helix where there's a hydrogen bond forming between the I and the I plus fourth amino acid, and basically uh, yeah, so basically for the I plus fourth amino acid, this hydrogen bond formation should be due to hydrophilic residues where you have a polar molecule there which can do that. But in case of it is shown here aspartame and serine, which are both hydrophilic in nature, which can form these covalent bonds. So in alpha helices, for forming those covalent bonds, you need hydrophilic residues because hydrogen bonds are required. But the amino acid side chains can be either hydrophobic or hydrophilic, and they can be localized in specific positions on the helix, forming amphipathic regions, which are basically not fully hydrophobic or neither fully hydrophilic natures in nature. So it can contain both type of residues. So in amphipathic helices, hydrophobic residues are positioned on one side. So these are all your hydrophobic, the yellow ones, and the hydrophilic residues are positioned on the uh, other side. So this is what an amphipathic helix looks like. So this is a top-down view, right? So this is a alpha helix that you are looking top-down. So one side of it is polar, and the other side of it is non-polar. Okay, so this is a particular protein which is tyrosine protein phosphatase C, and here are the amphipathic strands. So here are your polar molecules which are forming the hydrogen bonds to keep the alpha helix stable, and other than that, you have all the non-polar bonds, non-polar amino acid residues in and around those helix structures. Okay, so one thing is like that I'm going week after week, like I'm following that chronology. So some of the topics might seem a little bit jumbled if you just study in a continuous manner, because first we're talking about alignment, then we are going to chemical properties, then we are going back to alignments and so on, so on. So it's just how the course was designed, but it might look a little bit odd, but I'm following the chronology, so it should be fine. Okay, so we've talked about PAM-BLOSUM matrices, the scoring matrices that are there for sequence alignment to find consensus sequences. And there are also specific type of other matrices which are known as PSMs. So these are protein-specific scoring matrices. So the idea is that if it's just a match or a mismatch, it's very rigid. So if it's a binary outcome, it's very rigid, 0 or 1. So what they do is, in this case, they replace 0 and 1 with some probability value, right? So these are probabilistic values that you find in this matrix, and you build a matrix where the rows are 20 amino acid and the columns are positions in the sequence. Each cell contains a score representing how conserved that amino acid is that at that position, and you add pseudo counts to ensure that no probability is 0. So even if you do not find anything uh, there, you can add a little bit of uh, in terms of probability to not get it 0. So 0 will be very biased, right? So yeah, and another concept that is used here is basically the consensus concept. So when you're calculating the probability, is like out of 10 proteins, what is the amino acid at residue position I across 10? So if P is present in all, the probability will be 1. If it is present in 80%, the probability will be 0.8, and so on and so forth. So this is what consensus sequences do. So consensus sequence will tell you what is the most probable amino acid at that position, and it will create a sequence on that. And each position is defined by majority vote with respect to multiple sequence alignment. As I was saying, if you have multiple sequences aligned, the maximum number of amino acids in that position will be in the consensus sequence. Okay. Another similar type of concept is with respect to sequence logos. So basically, here also it's a display of multiple sequence alignment. If you have one particular, in this case, it is shown with respect to nucleotides, but it's the similar concept with respect to proteins and amino acids. The idea is that if you have the height of the letter will tell you relative in a relative manner how many times it is appearing in that position. So if you see this particular position, position 5, all of them are C, so the C is so big. And if you see this yellow one, there are a lot of things. There is C, there is G, there is T, there is A. So you can see it's very small here. So these are what are known as sequence logos. So these are designed to display the frequency of amino acids or bases at each position, and the relative height will tell you uh, in terms of the frequencies. Okay. Now, how we are talking about PSMs, we are talking about sequence logos. All of these relate to multiple sequence alignment, right? So if you have multiple sequences, what is the consensus sequence, or what is the probability that one particular amino acid appears in that position, and so on. Right? So, with respect to multiple sequence alignment, we can extend it to phylogenetic analysis as well. So, what is phylogenetics? So, a phylogenetic tree or a evolutionary tree, it's a graphical representation in terms of mathematical graphs uh, of how evolutionary history of biological sequences and allow us to visualize the evolutionary relationships between them. So basically, uh, there is a root of the tree, and there are nodes, leaf nodes of the tree, which are your terminal nodes. So these are your final species. So for example, here you can see D and E are having common ancestor. B and C are having a common ancestor. A is sharing a common ancestor with B and C, and so on and so forth. So this is how a phylogenetic tree is read, right? So there will be a root, there will be leaves, and the edges connecting the leaves will relate to a common ancestor. So this is how a typical phylogenetic tree looks like. You can have two types of phylogenetic trees where you have one is a rooted where there is a common root. So in this case, there is a common ancestor across all the organisms or protein sequences that you are looking at, right? In some cases, there is no common ancestor. It can be something like this. Uh, that will be unrooted, and in case of rooted, there will be one common ancestor, which is not present in unrooted. Okay. Okay. So these numbers, I think we have discussed a few problems based on this in the sessions that we had for these concepts. But questions like these can come, and just kindly remember the formula. So you can ask a rooted tree, basically, excuse me. Yeah. So if there's a rooted tree, if there's n elements, how many possible? Uh, sorry. Sorry for that. Yeah. So in rooted tree, how many possible uh, rooted trees are possible with respect to n child nodes? How many rooted unrooted trees are possible with respect to n child nodes? So these kind of questions can come, and you just have to apply the formula and write the answer. So do remember the formula. This has been covered in class, as well as I think we did a couple of questions in earlier sessions as well. On the basis of topology, if you can have there are two types. One is cladogram. It will only show the branching pattern and the evolutionary relationship, and it is unscaled. It does not show us. tell you a sense of time or sense of distance between two taxa. And phylogram will be representing evolutionary relationship by both showing the branching pattern and also the scale of the timeline or the scale of the distance between two whatever protein sequences or taxa or two organisms and so on. Okay. Okay, now how do you construct this phylogenetic trees? So the original concept comes from multiple sequence alignment, right? So after multiple sequence alignment, you have found some similarity. Now you want to construct in terms of the similarity of particular tree, which will tell you the evolutionary relationship between these sequences or these taxa or these organisms, whatever. So first you select a molecular marker. So in our case, it will be just give me a moment. Sorry for that. Okay, so yeah, first you need to select a molecular marker. So in our case, it will be some conserved region or unconserved region of a protein. You do multiple sequence alignment across different organisms, and then you select a model of evolution. So there are different models evolution models, as in like the PAM model or the Blossom model or whatever metrics that you're using. So all of them assume a different model of evolutionary relationship, right? So there are different probabilities, as we discussed for the Jukes Cantor and the Kimura model, that they are using different probabilities to represent different changes, right? So yeah, you can use a particular model, and if you are using protein sequences, we are almost using PAM or Blossom all the time. And from there, you construct the phylogenetic tree. That will depend on the dissimilarity between distances or similarity between distances. You need to quantify how much similarity is there or how much dissimilarity is there. And then you finally assess the reliability of the tree by resampling the initial sequence data to uh, generate multiple subsets of derived sequences, and you do bootstrapping as we have discussed earlier as well. Okay, now here comes the concept that you're constructing the phylogenetic tree on the basis of some sequence similarity or dissimilarity. Now, how do you quantify that? So a very basic way to quantify that would be to just see how many identical characters are there, and then you take the minimum sequence of length A or length B, and then you find the similarity index. Or you can do what you can do is basically take identical characters, number of identical characters by total length of the alignment, not the minimum sequence. So the next one is a little bit more robust than the first one. Right? So that is one simple way of doing that. Other ways of doing that would be to compute a distance between two different sequences based on similarities and dissimilarities. So, sorry. So, the distance is going to quantify the the dissimilarity or the similarity between two sequence, right? So, number of different positions uh, will be two. So there are two. So if you take these two examples, there are two positions where the alignments are differing. So if there are two positions, that is the lowest possible distance difference between two sequences. That is what is known as the Hamming distance between two strings. So the Hamming distance would be, if you align two strings, what are the number of differences, number of mismatches? That is simply the Hamming distance, which is two. There is something also called the edit distance or the Levenshtein distance. So what it does is basically will tell you how many number of edit operations you need to transform one sequence to another, right? So if you want to start from this sequence and go to this sequence, you can do a lot of there can be a lot of possibilities. So for example, from going to this, first you substitute T with G, then you delete C, then you insert another T, then you insert another T, and then you reach here. right? So that is one way of doing it where there will be four edit operations. In another case, you insert a G in the second position and then you substitute the C with the T. There are only two edit operations. The third one is another possibility with three edit operations. So the edit distance will be the minimum number of edit operations, which is the two edits, the second option. Okay, so Hamming distance will tell you how many differences are there. Edit distance is basically how many changes you have to make to move one sequence to match the other, right? So that is edit distance. Okay, so now we are going to finally come back to the structures and the chemical properties. I think we have covered most of the. Yeah, here we are going to mostly talk about the. Yeah, we have a lot of things left. I think I should hurry up a little bit. Okay. uh, okay. Um, yeah, coming back to the structure of the proteins. Again, secondary structure, as I was saying, uh, stabilized by hydrogen bonding between residue I and I plus 4. So it is present in transmembrane proteins and so on and so forth. I'm not going to go into details. Yeah, beta sheets are another set of secondary structure. So the beta sheet looks something like this with the side chains oriented so that they extend above and below the plane. So you have the residues, the side chains basically, facing either the opposite direction or the same direction. So you can have either anti-parallel strands. Basically, you can have two strands which are connected to each other by hydrogen bonds. These are your hydrogen bonds, and these are anti-parallel in nature. So one is going from 5 prime to 3 prime, the other is 3 prime to 5 prime. You can have parallel strands as well. So you can have similar structures like these in the uh, uh, side chains, the hydrophobic side chains or other side chains that are facing each other. So that would be parallel if they're facing opposite to each other. So this and this. So these are anti-parallel. And in a secondary structure, you can have both parallel and anti-parallel sheets, beta sheets, right? Uh, okay, so I'm not going to go into these smaller details, so I'm going to skip these. We have already covered these in great detail. So you have hydrogen bonding here for alpha helix between I and I plus 4. Here also you have hydrogen bonding between two parallel set of sequences of the same peptide bond. Okay, so a little bit about the amino acid bonds or the amide linkages. So it depends on how the R group is present along that amide bond, right? So there are a few degrees of freedom that are possible with respect to the R and this amide bond. So on the uh, basis of the angles of these degrees of freedom, we define two uh, angles, phi and psi, on the basis of which we make the Ramachandran plot, and then where we can predict alpha helices or beta sheets. So basically, yeah, there are amide bonds which limit rotation on these amides, and basically, yeah, you have these two set of angles. One is the psi angle with, and another is the phi. So this is the psi, and this is the phi. So the psi is between the carbonyl carbon and the uh, the polar hydrophobic side chain, and the phi angle is between the nitrogen and the hydrophobic side chain. So this is what is known as the beta carbon. So the alpha carbon is the one that on which the carbonyl group is attached. This is the beta carbon, and this is the nitrogen. So alpha carbon and beta carbon angle is psi. Beta carbon and nitrogen angle is phi. Right. So these are the two angles, and there are limits on rotation uh, on these two set of angles, and on the basis of that, we have different types of uh, rotation. So in this case, if you this is psi equal to 0 with respect to the alpha carbon and the carbon behind it, and if you move it by 180, it just rotates. And these are other different angles. So yeah, the psi angle is between alpha and carbon. So basically this one, sorry, this one. this is the alpha carbon and this is the other carbon. And for psi, it is N carbon alpha. So this is N and this is the alpha carbon. So between these two, so one is with respect to the N, one is respect to the carbon in carbon, right? So these are the two angles possible, and on the basis of that, you can have all sets of rotations with respect to negative 180 to positive 180, and you have this Ramachandran plot. So based on psi and phi. So this is for one particular enzyme for which the Ramachandran plot is plotted, and you can see there are some places where there is more density, areas like these, and some places where there is very almost nothing. So those are more improbable angles, and these are angles at which amino proteins are found in nature or in humans or in other organisms. Now, coming to ways by which you can determine structures of a protein, the most common way to do that would be X-ray crystallography, where you have uh, where you use scattering of X-rays and basically produce images of it, and then you use uh, yeah, where is it? I think I have not included here. Yeah, so basically from X-ray crystallography is where you can compute the structures of these proteins, but the resolution is typically about 10 angstrom. Uh, and the formula, if you remember, 2d sine theta equal to n lambda is the formula. Okay, so yeah. So this is basically a representation of it. So you give the X-rays, this is your crystal. This will scatter the X-rays, and then you collect the X-rays and make a 3D density map, electron density map out of that. So yeah. Okay, so on the base, now we're coming to different classifications of different proteins. So SCOP is one such standard of classification by taking into account both structural and evolutionary relationships into account. There can be family, there can be superfamily, there can be fold related families as well, right? So this is one set of classification. So there are three levels: fold, superfamily, and family for SCOP. For CAT, the classifications are class, architecture, topology, and homologous superfamily. So these are different levels of classification that you want to do, and with respect to the protein, and these are different databases that implement that. Right? Yeah. So this is kind of a comparison where you have in SCOP you have class, the corresponding for cat is class. You don't have architecture for SCOP. For fold, it's a topology in cat. Superfamily don't have in cat. We have family and we have domain again. So yeah. Okay. Now a little bit about what the structure does to the actual protein, right? So what it does is basically it brings two amino acids which are very far away from each other to close contacts, which are known as long range contacts or short range contacts even. So you can plot these contacts with respect to their positions, and that is what is given here. So, so this is the original sequence, but here there is a contact here and another contact here. So these are all representing different levels of contacts. It can be short range, medium range, and long range. Long range are the ones that are the most interesting ones. And yeah, you can quantify or classify these contact patterns by structural classes, and this can show different levels of long range contacts. So yeah. Okay, so the another important thing with respect to the hydrophobicity and the overall structure of the protein is how much of the solvent accessible surface area is there on the protein. It will be determined by the amount of polar residues that is deciding on the surface of the protein, and there are different algorithms and computational methods by which you can compute solvent accessible surface area with respect to uh, what is the sequence and what is the structure in that region. Uh, uh, okay, so these are different metrics I'm not going to go into, but some of these may come in exams, so please be careful. So buried net index is something that also quantifies in terms of like how much accessible area there is. So how much buried the protein, the amino acid residue is. You can also quantify in terms of long range order contacts and so on and so forth. Uh, yeah, yeah. And finally, we have the protein folding problem and predictions of protein folding with respect to computational and statistical methods. Right. So the most important and the commonly used um, computational method to predict the protein folding or the protein structure would be homology modeling. The idea is very simple. You have similar sequence of amino acids in two different proteins. You know the structure of one protein, so you can derive the structure of the next one. So you have a you have to find a homologous sequence, and the homologous sequence should have a structure associated with it. You can do homology modeling on the basis of that. So yeah, you have a target sequence, you identify a template, you do sequence alignment, and then you build a framework model, and then you evaluate the model. But the main part of it is to find a similar sequence. You can have ab initio modeling techniques as well, where you do not have any prior knowledge of the similar protein structure or anything like that, right? Okay. Uh, yeah. Okay. And after that, had discussed about different types of matrix by which uh, we can define the protein uh, structure, right? So you can have center of mass. So these are physical parameters, right? Center of mass, radius of gyration. Radius of gyration will tell you how compact a particular protein structure is. You can quantify with respect to contacts, and there are softwares like PDB param which can tell you all this information. And after that, we had also discussed different types of stability of the protein. So with respect to thermodynamic and other stabilities. So yeah, this is the heat equation that we had discussed again earlier. So Gibbs free energy should always be negative for exothermic and spontaneous processes. And for protein folding, most of the cases, the delta G is negative. So it is a more favorable reaction to fold the protein rather than have it unfolded. Okay. Yeah, and there is this two-stage transition model is like either the protein can be in folded state or either in folded or not in non-folded state. So their probability must be always equal to one. So yeah, it's a two-state transition model. Uh, yeah, these are other parameters that we had again earlier discussed, but I don't think these are so important, but still know that we can quantify these with respect to fluorescence intensity. But the most important part of it is part of the quantification of stability is with respect to the thermodynamics, right? So this formula, remember, delta G equal to negative RT ln K, and K is defined as FU by A, right? So if you have the proportion of unfolded protein, you can find the proportion of folded protein. You can find K. You can substitute K, and you already will be given R and T values, so you can find the delta G. So small problems like these can come in the examination as well. Okay. Uh, then at the secondary and tertiary structure level, we have different types of bonds that are forming, which are stabilizing these protein structures. It can be electrostatic bonds, which are going to be formed between two polar amino acids, possibly between positive and negative amino acids, which are known as salt bridges. You can have hydrogen bonds. You can have Van der Waals forces as well, and there can be other physical forces possible as well. Also, hydrophobic free energy is basically what proportion or what concentration of amino acid is present in water versus non-polar solvents like octanol. Like, say, for example, uh, you have more hydrophobic residues present, so your uh, hydrophobic free energy will be less because it will solve more in solvents, and if you have more hydrophilic residues and charged residues, your hydrophobic free energy will be much higher. Right. Okay. So I'm going to skip these things. Yeah, disulfide bond. So all of these we are talking with respect to stabilizing the protein structure, right? So yeah. So one thing is this thing had been mentioned in the class as well, and one question was there from the from this set as well. So hydrophobicity is the primary driver, right? 50% of hydrophobicity, hydrogen bonding is 27%, and so the rest of them are lower. So even though electrostatic and disulfide bonds look to be very important, the most important thing is hydrophobicity. That is what is controlling almost every aspect of the protein structure. Okay, Gibbs free energy and all these things I am not going to go into again. Okay, yeah, protein folding rate is also something that we had discussed, is basically how fast or how slow a protein can fold. That is going to be the folding rate. Again, which interactions will drive the protein folding? Again, we discussed hydrophobic will be the most important one, right? And this is the thermodynamic uh, information about protein folding. So at this particular temperature, the delta G fold is 0, and after this temperature, it increases. So basically, delta G should be negative for protein folding, right? So if you keep on increasing the temperature, after a certain temperature, the delta G will become positive, and the opposite reaction is going to happen, in which the folded protein will become unfolded. And in this region, it's going from opposite, unfolded to folded, because delta G is negative. So temperature is a very important aspect of it, and also the kinetics of it as well. So there's this Lewinthal paradox is basically needle in a haystack problem. The idea is that the protein will always fold in the most uh, stable state, but it's an astronomically large uh, sample space on which the protein can fold. The idea is that if you have a particular sequence, you can fold in like millions and millions of ways. How does protein find to fold in the most conformationally stable state? So this is what is known as Lewinthal's paradox, which is the needle in a haystack problem. Uh, yeah. Okay. Yeah. Okay. So now, with respect to the how fast the protein is folding, there are a lot of properties influencing that speed. Uh, Most importantly are your amino acids. Where and how are the amino acids distributed? Where are the hydrophobic residues? Where are the hydrophilic residues? And so on and so forth. Uh, in terms of contact orders, how much long range contact is there? How much other contacts are there? Multiple contact index measures that as well. So this is the Anfinson's theorem. So the idea is that environmental properties will contribute to specific 3D structure of the native protein, but the most important determinant of the folding structure is the amino acid sequence. That is the most important thing, and this person won a Nobel Prize uh, after conducting experiments showing denaturation and naturation, and basically these are the known as what in today's world the thermodynamic hypothesis of the protein folding, right? So the idea is that the amino acid sequence is the most important thing. On top of that, you will have different factors like topological order, long range contact, and so on and so forth. But the amino acid sequence is the most important thing on how fast the protein will fold, or where and how the protein will fold. In terms of, he showed it with respect to naturation and denaturation. Okay, and finally, I think these are the final topics with respect to. Yeah, I'm not going to cover the ones that has had been discussed yesterday. If you want me to cover what had been discussed yesterday, I can cover it in the next session, because it would have become repetitive, because yesterday only I discussed, today I'll discuss the same thing again. So if it is required, I can do it in the next session. Okay. Yeah, and the final topic for this day is computer-aided drug design. The idea is that you have a ligand and you have a receptor, and you want to build a ligand that can go and bind to the receptor and do its function, right? So there are five Lipinski's rule of five. Basically, your ligand molecule should actually mm, kind of follow these five conditions, three of these five conditions. I know. So I think two of these five conditions at least. So it should have a certain molecular mass. It should have high lipophilicity. It should have less than five hydrogen bond donors, 10 hydrogen bond acceptors, and molar refractivity should be around 40 to 130. So these are five conditions. And so basically what we are looking for is how the ligand will bind to the active site, which is also termed as the pose site in terms of CAD. Uh, and the pose will be the specific location, orientation, and conformation, right, of the ligand in the site. It's not only the residues at which the ligand is binding, it is also the conformation and the orientation at which the ligand is binding. And the best way to find it is using molecular docking techniques. So there are a lot of techniques, and I'm going to discuss this, but yeah, molecular docking it's a computational technique that you're trying to use to find how what kind of affinity is there between your desired ligand and the receptor of interest, and you quantify this on the basis of delta G, like how much free energy will be released where when this uh, binding happens actually in the cell or in the system. Somebody asked it. Okay. Yeah, and yeah, the most important aspect of it, the most important application of it is on with respect to drug development. So you are developing a drug which can go and bind to a particular receptor and slow down the effect of that receptor. A most common thing of it is targeting insulin receptors uh, using drugs, right? So, and the idea is that you're designing a drug and that should go and bind to a receptor, and you're trying to model this binding of the receptor with computational techniques. Molecular docking is the most common of them. So this is the general workflow. So you have a compound library. So you have a set of compounds that you think are good enough. Then you classify the compound based on docking scores. And based on that, you can classify the compound as active and inactive. You train a compound activity classifier. Then you dock the selected, and basically you get the positive and the negative hits from this classifier, and then you dock the positive hits into the target protein. And basically, if the docking is successful, you get those proteins and synthesize those compounds and evaluate experimentally in cell lines and so on. Okay, so the principle of molecular docking is that uh, it uses basically uh, yeah, so basically it uses a computational method to tell you what is going to be the affinity between your ligand and your receptor. Uh, uh, yeah, so that it can be yeah. So to establish a best interaction and imaginative sampling of all possible poses of the ligand in the specified groove is required to obtain optimal binding geometry, which then decides the best interaction. So all the possible poses as in the location at which the ligand is going to bind, and also the orientation of the and the conformation of the ligand at the position. At what position, location, and position, what is the best pose for this particular ligand to go and bind to that receptor, and what are the different energy delta G's that you get for different sets of binding. So based on that, molecular docking works. Right? So again, the major steps are: first you select the target, then you select the ligand, then you do the docking, and then you evaluate the docking. The docking evaluation can be done with respect in with respect to computational methods using scoring functions, or some wet lab techniques like NMRs or X-ray crystallography. If you can have so if you can isolate this bound complex of your ligand and your receptor from an experimentally conducted experiment, and you take uh, that compound out, you crystallize that compound, and you do NMR spectroscopy or X-ray crystallography to find if it is correct or not. Okay. So the important part of it is finding the target, finding the ligand and preparation of the ligand. So for that, we do virtual screening. So we take thousands of compounds and we screen those compounds based on different conditions. So in silico screening, uh, we do basically with respect to all the data available for that particular compound. But for wet lab techniques, you can use you can use again in a cell line. You can grow a culture and you can try different compounds and see which of the compound works best, and then you take those set and then you refine a smaller set out of that in using computational techniques and do do that. So there are different types of virtual screening. One is receptor based, so compounds that will be similar, which will be complementary to the receptor. It can be ligand based also, which is basically complementary to the ligand. So the idea would be receptor-based screening would be done when you have a ligand ready and you want to find the receptor against it, and the opposite will be done when you have a receptor, you have a known receptor, and you have to find ligand. So that is the more common case when you know which receptor to target and you do not have a ligand to target with respect to. Right? Uh, yeah, and here also the delta G is the most important thing. Uh, on the basis of the delta G, you're going to classify if this particular compound will rank higher or lower in your screening method. Okay. Yeah, there are other factors also involved: solvation, dissolution factors, and delta G is basically when you're doing the docking. It is considering a lot of things. It is considering the solvation, it is considering motion, configuration, and interaction. And all of these delta G's are going combining and giving you the final combination combined delta G of the binding. So the molecular docking program will calculate across all these. Okay. Okay, I think these are again uh, screening. Yeah. So one such screening technique is QSAR. So this is one of the most commonly used screening technique. Uh, yeah. So this again, as I was saying, this is the virtual screening workflow. You start with a target. You so it can be structure based virtual screening or it can be ligand based. So basically, this is the receptor based and this is the ligand based. So you take all the possible uh, uh, ligands here for ligand based screening, and you take all the possible uh, receptors, and then you run it through some machine learning model in this case, and then you see which one is the most similar and the most appropriate one. Okay. And QSAR is basically a specific technique which uses the biological activity of the compound, which is in this case they have used IC50, which is the half-life of the uh, of the drug inside your body. So how much time the drug is active and how much after time it becomes inactive. So that is what IC50 tells you. So if you have a compound like this, so if you have several compounds, you're doing virtual screening, right? You have several compounds, you have several IC50 values, you have several molecular descriptors. Right, it's mass, it's amino acid composition, how much of it is hydrophobic, how much of it is hydrophilic, and so on and so forth. So you can make a linear regression model out of that. So your this IC50 value you can predict with respect to uh, all the uh, chemoinformatic values that you have, molecular descriptors that you have, and then you can find which act of the molecular descriptor is actually predicting the best IC50 value, and then you use that descriptor to screen for the different compounds. Right. So this is a more of a linear regression based method where you uh, um, want to kind of predict the molecular activity with respect to IC50 or binding affinity with respect to the structural and physiochemical properties. And what it does is it establishes quantitative relationships between descriptors and the target property capable of predicting activities of novel compounds. So if you have the K1, K2, Kn of these these. So if you have a new compound, you can predict the IC50 value from this regression technique. So yeah, I think I think this was all till week 10. I've not discussed week 11 as I was saying, because again I discussed this it last day only. It would have sounded very repetitive, and I think we are also out of time. So if required, I can discuss this in the next session. Please let me know if it is required. Okay, any questions from anywhere?
Sir, it would be helpful if we get any of a formula sheet of NAT type base question. Yeah. Because it's very difficult to uh, filter what is useful and what is not.
Uh, so, can you repeat again what exactly you want?
I was asking that for NAT numerical answer type questions,
Okay.
is there any kind of formula sheet available or overview of the formulas in all of the week?
I don't think so, no. You
Okay,
have
it's
to
alright, sir.
you have to base your preparation on the assignment problems, and uh,
Okay, sir.
so see, the NPTEL examination is not very drastically difficult. It is going to be almost on the same level as you have seen the assignments do, slightly more than that maybe, but the questions are not going to be vastly like very difficult from what the assignment problems are. The type of question will be very similar. So if you cover all the like 10, 12 weeks of assignments that has been done, I think you're good to go. I don't think we'll have to worry about formula sheets and all. And
Yeah,
there are no formula
assignment
sheets like
questions
that.
are good. Assignment questions are good,
Yeah.
but I was a little bit worried about the exam.
No, I don't think it'll be that difficult or anything if you practice assignment questions well. And basically, are you given some other practice questions as well, other than the assignment ones?
Yes, AI-based questions we
Yeah,
are
yeah, yeah,
given. We are also
yeah.
given formative assessments also.
Yeah. You follow those, I think you'll be good to go. It's not going to be a problem. It's not the exam is not going to be drastically difficult or anything, so don't worry about that.
Okay, sir. Thank you, sir.
Okay. Yeah. The understanding is the important thing. If you have, so see, these kind of courses, like, yeah, you cannot expect like in 12 weeks you'll know full bioinformatics. That is not possible. So these kind of courses are mostly there to give you an idea about the field, what kind of work has been done on the field, and what kind of work can be done on the field, so that it gives you an interest and an intuitive understanding of the different concepts. And if you are interested in the future, you can go and pursue it. So the question paper setters are also not going to expect that you will know everything. So it's okay. Don't worry about it. The main thing is the understanding, and if you have gained some interest out of this course, if that is there, it's all it's good. And if for the exam, you just practice the assignment questions and the other questions provided to you, I think will be good enough.
Okay, so thank you so much, sir. And interest-wise, it was a very interesting course.
Okay.
Uh, as I was always keen in 12th class also, I was always keen to learn about what bioinformatics do and
Oh,
how
that's
does
good.
it
That's
work.
great. That's great.
Yeah.
The field has a very bright future, so yeah, if you want to continue, it's a very good field to continue.
Thank you, sir.
Okay. Okay, then I think if you have any other questions, or else we'll close the session.
Hello, sir.
Yeah.
Sir, I have a question, a little bit, and it is not inside from the lecture which you took today. Sir,
Okay.
it is about the two amino acids. One is pyrrolysine and one is selenocysteine. So I was reading about them,
Okay.
and I got to know that the codon, which code for those two
Hmm.
amino acids, are
Hmm.
just stop codons.
Yeah.
So
Yeah.
I'm actually looking for how they got detected and how they actually work. Like if there is a tRNA, if there is tRNA for those stop codons having those amino acids, then how are they working as stop codon? and so.
Okay. I do not have a correct answer to that. I'm sorry. I do not know the mechanism to that, but the examples that you are giving are very interesting one. Pyrolysin is a very well studied, I think bacterial, it's a bacterial
It's a
protein.
bacterial
Protein,
amino
yeah.
acid.
Yeah, yeah, yeah,
It has been found in those them.
yeah. I can take a look and see if I can find any mechanism and I can get back to you. But right now,
Mhm.
I don't know. I'm so sorry.
Actually, I read about them, but it
Yeah.
was quite confusing. Well, whatever is mentioned, it was
Okay.
quite confusing that there is serine on the tRNA, and then it gets changed, converted into selenocysteine.
Okay.
Then what is serine doing on the tRNA if there is a stop anticodon?
Okay.
So these are few confusions.
Okay. Okay. I'll come back to you next week because
Okay,
I do not have a mechanistic idea of how this is happening. So I
okay,
have
no
to come
problem.
back to you. Yeah. I'm sorry.
Okay, okay. Thank you, sir.
Yeah. Okay. Uh, if anything else, I have noted it. I'll come back to you next week because I think in next week's class also I don't think there's not very much to discuss. So if you have any other topics like these you want me to discuss, I can take it up.
Sure, sure, sir. Thank you so much, sir.
Yeah. Okay. Thank you guys for joining. See you next Tuesday.